Today our topic is linkage in chromosomal basis of inheritance. So first you must know what is linkage. That is the linkage, the genes only determines the character. So the genes is responsible for the characters of an individual and how it is carried, on what it is carried. It is carried on the chromosomes. So when the genes are present in different chromosomes, they assert independently according to Mendel's laws of independent assertment. So we had seen the laws, 3 laws, in that the genes will be assert independently as Mendel's view. But some of the genetic characters did not assert. This was done by some of the biologists who do the experiment on the Mendel's law of independence, but they had get an different view that some of the genes are not assert, not separate independently. So the first case, that is, it is reported in sweet pea, Lathyrus odoratus, by William Baxman and Reginald C. Punnett in 1906. So the linkage was given by, that is, with an experiment, they proved it that Mendel's laws of independent assertment is not applicable to all the genes. Okay, so for that, in Lathyrus odoratus, they took the purple color flowers and long pollen grains. In previous version of the books, that is in old books, they give blue flower and long pollen. So both are correct only. So blue flower or purple flower, long pollen grains, okay, is crossed with red flowers and round pollen. Okay, this is same. Only in purple they change, that is in old books it will be blue color, but in new book it is purple color. So they cross these two characters. In F1 generation, they got an heterozygous condition, purple flower with long pollen. Okay, so that is the dominant flower is exposed. Okay, after that, the F1 is crossed with the test parent, that is recessive parent, that is test cross is applicable. The F1 is crossed with the recessive parent, test cross. And in F2, as per Mendel's ratio, the test cross contain 1:1:1:1:1. This is the ratio has to come. When the F1 is crossed with the recessive parent in test cross, that is the standard ratio has to come. 1:1:1:1:1. But here in this experiment, we didn't get the expected ratio 1:1:1:1:1. So, okay. So, these genes do not allow themselves to separate. So, some of the genes, as I said in the beginning, the genes they do not assert independently. So, some of the genes are not separated. They remain and they cannot independently separate. So, the type of tendency of genes to stay together during separation of chromosome is called linkage. Listen students, the last point, that this type of tendency of genes to stay together during separation of chromosome is called linkage. And if they ask the definition of linkage or what is linkage, you have to give this last 2 points. Instead of putting this type, the type of tendency of genes to stay together during separation of chromosome is called linkage. That is, some of the genes are together and they won't separate during chromosomal separation also. That is called linkage. Hope you understood. Next we will see a example, the experiment what they had done by Baxman and Punnett. So as I said, this is from an old book I had taken. Okay, this is the old book and what I also studied this type only. For you only they had changed. So blue flower with round pollen, capital B, capital L is dominant, is crossed with red flower, round pollen, small b, small l, and the gametes are separated as capital B, capital L and small b, small l. So in F1 generation they get blue flower, long pollen, that is capital B, small b, capital L, small l, that is blue flower, long pollen, the dominant character. Now the F1 is test crossed, that is the test cross means the F1 is crossed with the recessive parent. Okay, that is blue flower, long pollen in heterozygous condition is crossed with recessive parent. This is the recessive parent, small b, small l, that is red flower, round pollen. So the gametes are separated here as capital B, capital L, capital B, small b, capital L, small b, small l. This is from the F1 generation you get the gametes, and from the recessive parent you get small b, small l. Now you are crossing it. So the first one you get capital B, small b, capital L, small l, that is blue long. And the next one capital B, small l, small l, that is blue round, the second one. And that is third one is small b, capital L, small l, red long. Okay, and last one is small b, small l, small l, round, that is red round type. So these are the things, and the expected ratio is this one, 1:1:1:1. Instead of that you are getting the ratio as 7:, that is 7 blue long is to 1 blue round is to red long, that is 1 red long is to 7 red round. And this one, that is the 7 blue long and 7 red, that is red round, are the parental combination. Okay, parental combination. So if they are together they produces more. Okay, if they are together, their capacity, their percentage, their frequency is more, the recombination is more, because they have more number of linked genes. But in 6, that is 6:6, that is recombinant, newly formed from the parents, that is blue flower, long pollen into red flower, long pollen, that is round pollen. During that the offsprings has been appeared. So they are in much, because there are so many unlinked genes which are not linked. So they separate independently, but because of that they have, the frequency is less. Their frequency is 12%, 6 plus 6, 12%. So this only, that is the linked genes are together, they give the frequency more. The recombinant frequency is more, and because of the linked genes are present together. So that is called linkage. If they are apart, the genes are apart, they are known as unlinked genes. So they are apart. So we will see it clearly what is linked gene. So I had given a picture here, that is gene 1 and gene 2. So here I had given a picture, gene 1 and gene 2, and you can see how they closely seen. So this is the closely seen position, how they have been closely seen in the position, that is in the locus. They are very nearer, so they cannot able to separate. So that means they cannot able to separate, means they are called linked genes. The characters are linked together. Now I am giving the definition. The genes located close together on the same chromosome and inherited together are called linked genes. So what I had explained, you got the idea from the definition, no? The genes are close together on the chromosome. So this is the chromosome. The genes are very close, as you are seeing in the picture, they are very closed. And after they are transmitting to the another generation also, or another that is during meiosis or during that is gamete formation also, these genes won't separate. They will be remain together. That is called linked genes. Next, unlinked genes. Unlinked, you can see the gene that is here, one gene, that is gene 1, and here gene 2, and you have seen the gap here between the gene 1 and gene 2. Okay, because they are apart, you can see the gap. That means how the genes will be having inheritance of these two. So they are called unlinked genes, and this phenomena is called as synteny, and these genes are also called as syntenic genes. The two genes that are sufficiently far apart on the same chromosome are called synteny genes or unlinked genes, and this condition is known as synteny genes. That is, here one gene, you can see the gap, the distance between the two genes. So they are far apart, they are present in the same chromosome only. This is the same chromosome only. They are, one is on the top and one is on the bottom. So that means the genes aren't linked. So they can separate easily. They can separate easily. That type of genes are called unlinked genes or syntenic genes. And this condition is called synteny. And based on this frequency value, it is called, that is due to the recombination only you are getting a value. That value is called recombination frequency. If the value is more, recombination frequency, that is more than 50%, more than 50%, two genes are unlinked. If the frequency is less than 50%, it is linked. So closely linked genes are called linked genes. Okay. If they are located, that is strong linked genes that are closely linked, that is this type is called, so this type is called closely linked genes, and we can say it as strongly linked genes also, strong linkage also. And here they are apart. In this they are apart. They are far away. Each gene is far away in the same chromosome, and it is called closely opposite, that is strongly opposite, weakly linked genes. So based on the frequency, if it is more than 50%, then they are called unlinked genes, and the linkage is there is weak. If they are less than, that is the frequency value is less than 50%, it is strong linkage, and it has linked genes. Okay. Thank you for watching the video. If you have any doubts, post your doubts in the comment box. If you like the video, give a thumbs up, share and subscribe to Science Easy Tech channel.